So we're doing meiosis in 20 minutes, hopefully. I thought we'd just do a mitosis, meiosis summary first, because people bring in their GCSE knowledge, and it's too vague or sometimes wrong. So people say mitosis takes place everywhere. Meiosis is what sperm cells do, so it's completely wrong when you say that. We need to think A level, and in our syllabus, we've met mitosis in terms of the clonal expansion of the activated B and T cells in our immune system. So I get an army of plasma cells and I get an army of cytotoxic T cells and memory T cells and memory B cells. We're yet to meet cardiomyocytes. They're in topic 8, but they're a stem cell found in the heart, and they will like replace heart tissue. The hearts can grow if you do lots of aerobic exercise for years at at a very high high Olympic standard or something. Then hearts can grow in size. Whereas meiosis, sperm cells and egg cells, they are products of meiosis. So we're talking about these, we call them germline stem cells. So germline just means that they're passed on to the next generation. Stem cells can continually divide, and these line the testes, or they were in the ovaries when you were a fetus in a female, and hence why it's easy to talk about testes, because these are the cells that are just churning out sperm cells all the time post-puberty. Chromosome number in a human, there's 46 chromosomes, 23 pairs. We call those cells diploid because chromosomes are in pairs. The diploid number for a human cell is 46. It's just, we always count the number of chromosomes. If you use the word haploid or diploid, we just count the number of chromosomes. So don't bring in pairs or anything like that. But diploid, the word means, and they are in pairs. I've got 46 chromosomes and there would be 23 pairs. But the diploid number is 46. Not everything is like a human. Chimpanzees have got 48 chromosomes, and other mammals have got different numbers. There's types of plants that have got hundreds of chromosomes. In meiosis, we halve the number of chromosomes. The cells are called haploid. And then why needed? GCSE, we say growth and repair. It's kind of similar, apart from with my B and T lymphocytes, I'm trying to get an army of them. Would you call that growth? I'm trying to grow an army. Because they're kind of separate cells, I'd be just a bit more clear and say, to increase the number of cells if it's related to B and T lymphocytes. And meiosis, it's the same as GCSE. I need to halve the number of chromosomes so that at fertilization I can restore the number. And crucially, it gives me an opportunity to get a unique combination of alleles of genes. So I can mix maternal, paternal alleles of genes. And that's what we're going to look at now, because these words, maternal and paternal, and homologous chromosomes and chromatids, sister chromatids, are what people get confused with. So here we have a pair of chromosomes. Let's just label them first of all. So this black splodges in the center. This is our centromeres. I'm pointing to one here, but there are two. There's one at the top and one at the bottom. These chromatids are held together by their centromeres. These here are sister chromatids. Okay, so one chromatid is a copy of the other chromatid. Whereas these, this one and this one, these are called non-sister chromatids. However, they carry the same genes. We call them non-sister chromatids because they have different alleles of genes, or they may have different alleles of genes. So these are homologous chromosomes. So homologous means that they have the same genes. So they have the same genes. However, they may have different alleles. One chromatid might have brown-eyed allele, and the other one has got a blue-eyed allele. So, in terms of where I get these from, just trying to find a different color. Let's get a purple or something. Yeah, these are, let's call this one the maternal copy. The opposite of maternal is the paternal copy. So I've inherited one of these from each of my parents. It's not a male and female chromosome. They're not sex chromosomes. This is just one's inherited from my mother, the maternal copy, and the other one's inherited from my father, the paternal copy. They carry the same genes at the same loci. However, the alleles of genes may be different. So most of the genes will be the same because most genes, there are not alleles of them. They are called monoalleic. So, if there's a DNA polymerase gene at this position here, at that locus, it's going to be the same because this is a copy of it on that strand. And the same goes for, it's going to be the same on the maternal copy because there's no advantage for having a defective DNA polymerase gene. However, further down at this locus, this might be saying you're going to have freckles. However, on the maternal copy, it might be, no, you're not having freckles. However, freckling is dominant, and therefore the child would have freckles anywhere on the skin exposed to UV light. Okay, so that's just homologous chromosome terminology. When we talk about how meiosis works, we need to be using these terms: maternal, paternal, sister chromatids, non-sister chromatids, and we're going to still like collect the spindle to the centromeres. These chromatids are going to be pulled apart from each other, etc. So let's move on to the next bit, which is just recognizing metaphase, because metaphase is like the key stage for me. We don't need to know the difference between metaphase 1 and metaphase 2 in meiosis. However, that's according to the syllabus. I still think it's well worth understanding what's going on because it makes a bit of sense. So one of these is showing metaphase in mitosis, and two of them are metaphases in meiosis. So let's have a look at this first one. So this is all the chromosomes are aligned on the equator. Spindles is going to attach. It's a diploid cell. Half are going to go to the North Pole, half are going to go to the South Pole. This is classic mitosis. And I know it's mitosis because it's a diploid cell. However, this one over here is also diploid. This one is haploid. So the end one on the right hand side must be meiosis. Okay. But yeah, this is the arrangement via the equator tells me if it's mitosis or meiosis. And this one's, they are on the equator. So this only happens when the cell is diploid on the equator. That means mitosis. This one over here is a haploid cell, but it's on the equator. That just tells me it's the second metaphase. So we can finish this one off. So mitosis, diploid, yeah, all chromosomes, I'm going to put CH dot for chromosomes, are on equator. Sometimes called the metaphase plate. Okay, so that's mitosis. I need to go to explain, describe and explain every single bit of mitosis. Meiosis, we only really want to understand it in terms of how do I mix the alleles up and what happens when it goes wrong. So yeah, so this is meiosis. It's meiosis 1. It's the first metaphase in meiosis. There's the equator, and we can see that the chromosome pairs are next to each other, opposite each other, either side of the equator. And this is where we can talk about independent segregation, because if this is the paternal copy and the maternal copy, that is random. What side of the equator you are on is random. So I've got a maternal and paternal the other way around on the other chromosome, and therefore when I've got 23 pairs in a human cell doing this, then it is 2 to the power of 23, the number of pairs. That's the number of different possibilities that you can get. 2 to the power of the chromosome pair number. In a human, it's whatever, 5 million something or other from memory. It'd be impressive if I knew the number from memory exactly, but I'm not that impressive. Anyway, meiosis one. So we're going to say that the maternal and paternal copy. So this is where I say that, my maternal and paternal chromosomes or opposite each other. So I'm scribbling this down a bit. Either side of the equator. And position is random. Either side of the equator is random. So they do have to pair up. Position is random, and therefore this equals independent segregation. Independent segregation. And it gives us this mixing of paternal and maternal chromosomes in the gamete that is made. So, spindle's going to attach, I've got my centrioles here. Spindle's going to attach to the centromeres on one side. And when they retract, it's going to pull entire chromosomes made of sister chromatids. They're going to be pulled to opposite poles. And then the cell's going to do cytokinesis and split. And then I'm going to be left with two cells that look like this. Apart from, the top cell would be paternal and maternal of these chromosomes. These are no longer pairs of chromosomes. And the cell that I'm not showing would be the maternal and the paternal copy. Okay, so this is meiosis 2. Metaphase 2. Meiosis, it's Roman numerals 2. Yeah, and I know it's meiosis because the cell is haploid. And that's really it. It's not that interesting, this now. It's just like mitosis. We're going to have, our centrioles again that attach to the spindle, except they attach to both sides of each chromosome, so they're binding to both of the centromeres, and therefore we're going to separate the centromeres, and therefore the chromatids are going to separate, and therefore we are going to get our cells that have just got a single chromosome rather than a chromosome made of chromatids. Good. Right, I'm 12 minutes in, so I better speed up a bit. Otherwise, you'll be telling me off for going over time. So independent segregation, make sure you can write 2 marks about it, and you talk about maternal, paternal chromosomes. The one that's more complex, but also there's more marks for potentially, is when they cross over. So this occurs, and you don't need to know when this occurs. This occurs in prophase 1. So before they've gone onto the equator, the pairs of chromosomes come together and they synapse. Okay, so to show this, so I can say that that chromosome pairs synapse, or ideally form a bivalent. Bi means two, valent means form. It's in like a two form version. Okay, and this occurs between I can say chromosome pairs, however, not the X and Y. It'll occur between the X and the X. It doesn't occur between X and Y. Okay, so between the chromosome pairs, we're talking therefore mainly about the autosomes. These are the non-sex chromosomes. Okay, and you can see that these chromosomes I've got 2 crossover points there and there. And I can describe this. I can say that the chromatid from the maternal chromosome wraps around the chromatid from the paternal chromosome. Again, it's all a mouthful, and it's a bit of a nightmare. It's therefore why knowing the word chiasma saves you saying all that. Chiasma form. Yeah. So this is just a point of contact between non-sister chromatids. Remember from the previous stuff I said when I introed this, non-sister chromatids. Yeah, of a homologous pair though. And they're going to break and swap over. So the same lengths of DNA exchanged. If you're doing this correctly, we sometimes do this incorrectly, and different lengths are exchanged, but that's a later year 13 topic. So the same lengths of DNA exchange, and therefore we are mixing alleles of genes between maternal or paternal chromosomes. Okay, so just to kind of pop a little example on here, let's put gene A there. This one's got a gene A there, and it's got a gene um, big B right at the end over here. So and the other its pair, let's make this person heterozygous for both the genes, so they have a little a and a little b. So after they've done meiosis, one of the gametes is going to have the genotype big A, big B. So there's going to be a sperm made, and it's going to have basically this line here is going to end up in its cell. It's going to separate from its chromatid. It's going to end up in in a cell with the rest of the chromosomes. However, if I'm just focusing on these two genes, I'm going to have a big A, big B gamete. Because we only have one copy of the chromosomes. The chromatid over here that's not undergone crossing over, like this one's not undergone crossing over, I'm going to get a little A, little B gamete. There's a little sperm with a little A and a little B. However, have I picked a bad example here? I'm trying to think. Yeah, I haven't, because if I swap the thing over, yeah, by putting, I need to put. I'm just going to put a third gene on. I have messed up, but I am doing this a bit on the fly. Sorry, I'm going to put gene C here as well. So this would be gamete A, B, C. I'm now showing three genes in it, and let's put a little C on this side. C's not the best letter either, but I'm winging it. I'm going to wing it. Yeah. So that's got A, B, little A, little A, little A, little B, and little C as. If we do a crossing over here, well, there'll be a little B on this one originally and a big B on this one, but they're going to swap over. However, it's still going to be a big C here. All that's going to happen is that I'm going to make gametes that, well, I'm going to cross over. Take, I should just colour it in purple. Let's take this one here after it's crossed over. So it's going to take, I'm going to take the purple arm, I'm going to swap it for my yellow. And this is the one I'm crossed over with there. So it's going to end up with the little a from the other chromosome. It's going to end up with, it's going to keep, well, it's going to get the little b as well, but it's going to keep the big C because that's not been involved in crossing over. So again, just to show a bit clearly, so here on my purple chromosome is also going to be a big C. It's in the same place. It's a copy of the other chromosome. So this strand here is chromatid here is a copy of this chromatid here. However, I'm going to be crossing over the big A with the little a at the top because that section is going to be swapped over here, and my little b's are going to cross over as well. So I end up with a little a, little a, little b, little a, little b, big C gamete. And I also will end up with a big A from the other chromatid, a big B from the chromatid, and a small c because the small c hasn't been involved in crossing over because it's a copy of the other one. I've done that reasonably badly, okay? However, the thing to take away is that it's this bit here. That the same lengths of DNA are exchanged and we mix the alleles of genes. All this, how we get these gametes, you will do again. It's like after half term, it's not that far off when we do about, what happens when this parent mates with this parent, what's the probability of this child having brown eyes and freckles, and we focus on two genes, and then crossing over has an effect on the outcomes of those. So don't worry about my ABC thing. If it's made a bit of sense, brilliant. If it's not, don't worry too much. It's not going to matter in the upcoming assessment anyway. And I'm sure I'll be asked to do another meiosis video focusing just on that in relation to inheritance later in the year. Right, I'm already at 20 minutes. I do apologise, but I can do this a little bit faster, I hope. You need to be aware of a large-scale mutation. This one's called a non-disjunction. And you can see, all right, yeah, my spindle fibers haven't attached to one side of the chromosome. Okay, so this blue one's gonna, the chromatids are going to separate. On this one, the chromatids are not going to separate. So you're going to end up with a missing chromosome in one cell and an extra chromosome in the other cell. This diagram here is just showing that we're going to be meeting these problems later on in year 13. So again, this is when I've got crossing over in the wrong place. So I would end up with a longer chromosome and a shorter chromosome. If you can imagine that this is going to get that bit, it's going to be really long. So I'm going to end up with something that's all that red length, and this one's going to end up with like a little short arm afterwards. Yeah, it means that I'm going to basically lose genes from this. So we'll be meeting about that later, but it's just knowing that this can go wrong, and this can happen as well when you get crossing over between non-homologous chromosomes. This is the X chromosome, this is the Y chromosome, so this must be happening in a male, and therefore you can get genes moving from the X onto the Y chromosome. So you can get lots of like weird like sex determinism, so you can get like an XX male if the male causing genes end up going onto an X chromosome. Again, something we do later in the year. So I shall zip past that. It's just pointing out the non-disjunction is the one that you need to know about, where you don't get the separation of one of the chromosomes, as in the chromatids don't separate. So I should quickly just go through the cell cycle graph meiosis. Remember, we have these graphs where we have, I focus on mass of DNA in arbitrary units, so 0, 1, and 2. So we're going to start with 1 unit of DNA. We're going to go through interphase. Well, it goes through G1 first, where there's no, oops, just in that wrong straight away. So yeah, so that's where its growth, so its mass must increase. So let's increase its mass a bit. Then it goes, so that's G1. Then it goes through S, where it doesn't do anything in terms of growth, but it does copy its DNA. So it's just going to go across there. So that's the S phase. And then G2, it grows to full size. G2. Then in meiosis, it's organizing its chromosomes, and then right at the end, it does cytokinesis and it will halve in size. So this is cytokinesis. This is in sperm, by the way. It's different in eggs. And then in meiosis 2, it does just another organizational step, very much like mitosis, and then it will come down to here, and therefore that's our second cytokinesis, and therefore, every cell made is 0.5. It's like half the size of it was at the start of the cell cycle, or a quarter of the size it was when it got to full size. Okay, and it's just knowing that egg cells it's different doesn't really matter, but just to kind of zip through it quickly, it's going to be the same at the start. It goes along here. However, because I want to keep an egg cell large, one of the cells keeps most of the cytokines. So it's not an even cytokinesis. So you end up with a one going way down here and one going up here. So it looks like a bit like that, and then that's going to halve, and then this is going to like stay like that, and then that's going to halve down. So you end up with like one massive cell and three little cells for a egg cell. Whereas sperm, they just all become equally sized sperm cells. 25 minutes. So if I go further, so hopefully there was something useful there. Learn your terminology: homologous, crossing over, bivalent, chiasma, sister chromatids, non-sister chromatids, centromeres, centrioles, maternal, paternal, yeah, haploid, diploid. It is a lot of words to learn. Anyway, thanks for listening.